Good afternoon. I am Marina Loban, the creative inspirer of the MAGUS project. Today, we will talk about microscopes that are suitable for both students and professionals. Intensive use of equipment in classrooms and laboratories quickly renders microscopes inoperable, so reliable devices are needed here. Another problem faced by microscope owners is the inability to equip it in the future with devices that provide additional methods of contrast enhancement. I present to you the models of our basic line, the MAGUS Bio 230 and MAGUS Bio 250. These microscopes are ready to operate continuously for many years in the most intensive mode. The accessories expand the range of research possibilities in the future. The video will be long, but we will cover all the nuances of the microscopes. By the end of the video, you will definitely be able to choose the model that suits you best. And perhaps you will want to get acquainted with our research-level microscopes. The microscopes are built on the same stand, so at first glance, they may seem identical. However, all 8 models differ from each other. Firstly, the MAGUS Bio 230 and Bio 250 series are equipped with different optics. The microscopes in the Bio 230 series are equipped with achromat objectives and eyepieces with a field of view of 18 millimeters. In the Bio 250 series microscopes, the objectives are plan achromats and the eyepieces have a field of view of 22 millimeters. This is the second distinction. Each series offers models with binocular and trinocular visual attachments. The third option is the light sources. In this range, you can choose between a halogen lamp or an LED. In total, we see 8 models that differ in optical correction, visual attachment, and light source. Now let's take a closer look at the listed differences and discuss what unites these models and for what purposes each model is suitable. First, we will determine the degree of aberration correction of the objectives. For educational purposes, the Bio 230 series microscopes with achromatic objectives are suitable. These objectives correct spherical aberration for 1 wavelength of light, chromatic aberration for 2 wavelengths, and correct field curvature by 65% when using eyepieces with a field of view of 18 mm. For laboratory work, it is preferable to choose planochromatic objectives, which correct chromatic and spherical aberration, just like achromats, but they correct field distortion by 90% when using eyepieces with a field of view of 22 mm. This is the Bio 250 series. Just imagine the difference. A flat image across the entire field of view with a diameter of 22 mm or 18 mm, with distortion at the edges. We recommend choosing the Bio 250 series with planochromats, especially if you plan to work with a camera. The optics have been decided. Now we determine the type of visual attachment. We choose either a binocular model or a trinocular model. The microscopes can be connected to a computer or television using a camera. The cameras transmit images to the screen in real time, for example, during a seminar. They can also take photographs, record videos, and save files. If you plan to connect the microscope to a computer or television, choose a trinocular model. If you need a microscope only for visual observation through the eyepieces, we recommend saving money and opting for a binocular model. In the third step, we choose the type of light source, halogen lamp or LED. The halogen lamp illuminates the object with a stream of warm white light. The researcher's eyes do not strain. Therefore, if you will be working with a microscope all day, choose this light source. The drawback of the halogen lamp. It heats up, requires replacement, and changes color temperature with changes in brightness, which can cause discomfort when working with the camera. The advantage of the LED is that it consumes less energy, provides bright white light, and lasts longer than a halogen lamp. The LED does not change its color temperature when the brightness is adjusted. Therefore, the camera will not require white balance adjustment when changing lenses. The drawback of the LED is its narrow spectrum, which is unfamiliar to the eyes. Therefore, if the microscope is needed for short periods during the day, or if shooting with different objectives is anticipated, choose the LED. The choice of a specific model, MAGUS Bio 230 or Bio 250, depends on the user's specific tasks. To achieve results and work comfortably, determine the level of optics, the type of visual attachment, and the type of light source. Next, we will consider the general characteristics of the microscopes from these 2 series and their design features. The metal stand and durable materials of the moving parts ensure mechanical stability. Even after many years of intensive use, the microscope will continue to operate smoothly. We are confident in this and confirm it with our extended 5-year warranty. The Infinity Optics allows for the installation of additional modules, such as a fluorescent attachment, which can be purchased separately. Adjusting the lighting according to the killer will minimize errors and enhance the resolving power of the lenses. This results in an accurate image with detail at the limit of resolution for each lens. The The field of view of 18 min or 22 mm creates comfortable conditions for observation. The BIO 230 and 250 series microscopes are designed for observation in bright field under transmitted light. Additional accessories will expand the contrast methods: dark field, polarized light, and phase contrast method. A reflected light attachment is used for work with the luminescence method. However, we recommend purchasing a special fluorescent microscope, which is better suited for these tasks. Now, let's turn our attention to the design features of the microscopes. The Zeden top type visual attachment with rotating tubes around the axis. The user selects the upper or lower position of the attachment depending on their height. The interpupillary distance varies from 48 to 75 millimeters, up to 75 millimeters. Therefore, the microscope is suitable for both adults and teenagers. The diopter adjustment on the left tube helps to calibrate the diopters according to the user's vision. The eye cups protect against side light. In the trinocular model, this knob switches the light flow to the visualization channel. In one position of the knob, the light is directed to the eyepieces, and in the other position, it is directed to the third channel. This switching method ensures that the brightness of the light source is sufficient for use with any objective, not only for the bright field method, but also for the dark field and phase contrast methods. The second interesting property of such a light flux distribution is the absence of parasitic illumination. Only the image of the specimen is transmitted to the camera. Light entering the eyepieces from external sources will not pass into the visualization channel, and reflections from the ceiling light bulb will not ruin your photograph. To install the camera, we perform the following action. We connect the camera with the C-mount adapter, remove the dust cover from the visualization channel, install the adapter there, and secure it with a screw. The standard equipment of the microscope includes a C-mount adapter with 1x magnification, a camera with a small sensor on a single adapter will provide too small a field of view. So for such cameras, we recommend additionally purchasing C-mount adapters with magnifications of 0.5 or 0.65 times. To center the camera in the optical axis, there are 3 screws on the visualization channel. The standard equipment of the microscope includes 10x eyepieces. Additional eyepieces allow for an expanded magnification range of the microscope, as well as achieving useful magnification on each objective. The 10x eyepiece with a scale is used for measuring objects. If your research requires polarized light, you will appreciate the convenience of installing a simple polarization device. The polarizer is attached to the collector. To install the analyzer in the optical path, it will not be necessary to remove the visual attachment. The analyzer is installed in a special slot in the stand above the objective revolver. Pay attention to the revolving nosepiece. The revolving nosepiece of the MAGUS Bio 230 and Bio 250 microscopes allows for the installation of 5 objectives. The standard equipment of the microscopes includes the most popular objectives with magnifications of 4x, 10x, 40x, and 100x. Additionally, 20x or 60x objectives can be installed in the free slot, while the other objectives remain in the revolving nosepiece. The revolving nosepiece is directed towards the stand. The space above the stage is free. This means that the operator can see both the object of study and the working objective. Let's consider the objectives themselves. The objectives are designed for a tube length of infinity, with a parfocal height of 45 mm. As I mentioned at the beginning of the video, the microscopes in the Bio 230 and Bio 250 series differ in the degree of aberration correction of the lenses. Achromats and plan achromats. The objectives are interchangeable, so an additional plan achromat objective will fit into the Bio 230 microscopes. Look at the stage. The specimen is moved along the X and Y axes using this knob. The 2-coordinate stage does not have a protruding gear rack, which enhances the safety of the technician's work. The movements of the specimen are smooth thanks to the belt drive. The holder's foot is moved to the side and gently secures the specimen. The focusing mechanism moves the stage along the optical axis. For user convenience, the coarse and fine focusing knobs are coaxial, meaning they are on the same axis. The handles are located on both sides of the stand for the right and left hand. The range of movement of the stage is 21 mm, which ensures easy specimen replacement. The coarse focus locking mechanism is used for quick adjustment of the microscope when changing the specimen. After completing the coarse focus, it is recommended to turn the ring fully clockwise. This fixes the upper position of the stage. When changing the specimen, we raise the stage to the limit and focus only using the fine focus knobs. The mechanism for adjusting the stiffness of the coarse focus will help set a comfortable stiffness for each user. Another parameter of the focusing mechanism is the division value of the fine focus. On our microscope, it is 2 microns. This step is optimal for a student and routine microscope, as it provides both accuracy and speed of focusing. Next, let's examine the illumination system of the microscope. The ABA immersion condenser for bright field is installed in a spring mechanism under the stage. The dovetail mount easily allows for the replacement of the ABA condenser with a dark field or phase condenser. The spring mechanism of the horseshoe provides convenient centering with 2 screws and ensures the installation of additional condensers in the optical axis. The 3rd screw secures the condenser in the bracket. By centering the ABA condenser, you will be able to easily switch to other research methods without wasting time on recentering after changing the device. To illuminate using the Köhler method, it is necessary to center the condenser in the optical axis and position the condenser at a specific height relative to the object of study. The condenser is moved along the optical axis by rotating this knob. Another feature of the ABA condenser in the Bio 230 and Bio 250 models is the presence of a slot for installing a dark field or phase contrast slider. This design allows for quick and easy switching to other research methods without the need to replace the condenser. Above the light source is an adjustable dust diaphragm, which is also necessary for adjusting the illumination according to the Köhler method. Proper lighting adjustment allows for the maximum possible resolution on each objective. You will achieve uniform illumination across the entire field of view. The image of dust or glare will be removed, and you will observe a clear contrasting image of the object of study only. Changing the lamp is very simple. You just need to unscrew 1 screw. When replacing the lamp, remember to use gloves or a cloth. This will extend the lifespan of the lamp. Halogen lamps of 12 volts and 30 watts have high brightness and are available in stores. If you need to replace a LED, we recommend consulting a specialist. Pay attention to the stand. It features a convenient handle for carrying. The Bio 230 and Bio 250 microscopes are stylish, convenient, and reliable. We offer 8 different models and a wide range of options so that everyone can choose exactly what suits them. Let's summarize. First, we choose between the MAGUS Bio 230 with achromatic optics or the MAGUS Bio 250 with plan achromatic objectives. Next, you need to decide on the model within the series. The letter T in the model name indicates a trinocular visual attachment. The letter B refers to binocular. The final step is to choose the light source, halogen lamp or LED. The halogen lamp is not indicated in the model name. However, the LED is denoted by the letter L, which appears at the end of the model name. If, in addition to bright field, you need other contrast methods, we will help you select the appropriate accessories. Good luck with your work, colleagues.